Hi everyone. Today we're going to go over chapter 13.2, which reviews how to predict simple crosses. This goes over your textbook on pages 208 through 209 of your book. Now, we're going to basically take all the vocabulary we learned in chapter 13.1 and put it in context and use it for problem solving today. So this page you don't necessarily have to write down, but you have to be very familiar and very comfortable using all these terms. You need to be able to know what it means to be dominant or to be recessive in terms of what that means for a phenotype or genotype. Remember, recessive are traits and alleles that only will show their trait if both copies of the allele for a gene are recessive only, while dominant will show and present in that organism whether or not you have one copy of that allele or two copies of that allele for a gene. You also need to be familiar with the word phenotype or genotype and be comfortable using them because they're going to be used very frequently in problems. So phenotype, if you're asked to analyze a phenotype, it means you're being asked to analyze what that organism is showing, whether it's a certain color, whether it's they're tall or short, curly or straight, whatever trait you're looking at, that's phenotype that you're being asked to analyze. And if you're being asked to analyze genotypes, they're asking you to provide the alleles that are shown for a given gene. So you might represent those alleles using letters, so like 50% big B little b or 50% big B, or you might be using the terms heterozygote, homozygous dominant, homozygous recessive, things like that. And we're going to look at different ways these problems are used and how they get represented through letters and other things like that. Now, a big thing to understand is we use letters because it makes it way easier to track our predictions rather than writing out a super long DNA sequence. So when we're talking about, for example, eye color, at least in humans, an allele for eye color is blue or brown on this location of the chromosome, okay? But the actual DNA sequence, this is the real DNA sequence. And the actual difference between the brown color versus blue eye color is very little. It's basically one nucleotide different. However, when we represent that when doing crosses, we kind of always use a letter of the alphabet. And we identify which of these two is dominant, is a stronger trait that will mask the other by giving it a capital letter. Also, when we're identifying things that are of the same gene, different versions of an allele for the same gene, usually, though not always, there is an exception we're going to go over in the future, but usually we like to use the same letter of the alphabet. So in this case, the eye color gene, I might use B, so I'll use big B for brown and little b for blue to indicate that brown is dominant over blue. But I could also use any other one. I could have used E. So this could have been big E and this could have been little e. Okay? And then we represent those alleles through with those letters, and then that allows us to provide genotypes. So if I'm saying someone is homozygous, only has copies for the brown colored allele, then I would tell it's big B, or homozygous dominant. While if they have big B and little b, it doesn't matter which one comes first or which parent gave it to you. If you have one of each type, they're different. We call them heterozygotes. And generally speaking, when we represent their alleles, we always want to put the capital letter first. Big B, little b in this case. Or if it was E, big E, little e. Because we always want to have the letter that's going to match the phenotype come first. Because brown, the dominant one, is going to show, we want to put the big B first, not the little b first. And finally, we have an organism that has only the alleles for the recessive trait. So we're going to show that by saying they're little b for their genotype, or they might be called homozygous recessive in this case. Okay? And we use these letters because it allows us to predict the outcome of two parents. And we're going to learn how to use this. This is a Punnett square. And what we use for these Punnett squares is we put the alleles that the parents could potentially pass on to the child on each side of the Punnett square. And we use that to predict what the outcome will be in the inside of the square of their potential children. And each one of these squares represent, so there's 4 squares here. So this is a 1 out of 4 chance of this happening, 1 out of 4 chance of this happening, 1 out of 4 chance of this happening, 1 out of 4 chance of this happening. All right. And all this is just a representation of what's occurring during meiosis. Because remember, every organism has two versions of each chromosome, meaning two alleles for any given gene. And we are trying to predict the chances of each of the two alleles, or the alleles that each parent will pass on to their child. So for this one, because this individual is homozygous, what's going to end up happening is in the kid, this organism will always pass on the little p allele. Well, this organism, because they are homozygous for big P, they are always going to pass on the big P allele. So this parent and this parent, if they got together, would always create a child that is heterozygous with big P and little p coming together. But that might not be the case. Maybe the parent has a big P and a little p. And in this case, it would be a 50-50 shot of the child for this from this parent getting a big P or little p. And we would only know that by analyzing the original genotype of the parent. All right, and we do this with the Punnett square because it creates a really clean way of writing out the predictions of what's going to happen when two parents mate. It doesn't matter which side is the mother or father. You just pick one side for any parent. So this parent is the little p parent. This parent is the big P parent. Okay? For me personally, if I'm given an option to pick a letter, I usually try to pick a letter where the capital letter and the lowercase letter is very different, like big A, little a, rather than one where they look almost the same, like big P, little b, or big C, little c, or big S, little s. But as long as you can make sure it's really clear what the lowercase version is versus the uppercase version is, you do you. All right, so this is what we call a monohybrid cross, which is when you breed, you do an experimental breeding and you're doing a prediction of the outcome of a cross between two individuals for one gene. So it's not that these two individuals, this mother and father, don't have other genes. It's just that we happen to be only tracking the outcome of the offspring for this one gene, and we call that a monohybrid cross. Sometimes we represent the first generation produced from the cross as what we call the F1 generation. And then the children, when they have children, then they would be called the second generation, or F2. All right, so this is the parent generation, and then in this square represents the F1 generation from the first cross. And if these children have kids amongst themselves or with themselves, if this is a plant, for example, then that would be the F2 generation. So you can kind of see that in Mendel's work. So we have the parent generation, the original true breeding plants that bred together, their phenotypes, which are what they are, white versus purple, their genotypes, little p for the white flower, big P for the purple flower, because we know it's true breeding, it's always purple, which means its genes are always purple, only purple. But then the F1 generation between these two parents, because you're guaranteed one parent will only pass on the flower gene, the purple flower gene, and one parent will only pass on the white flower allele, then you're going to end up with hybrid children or heterozygous children with a flower allele and a purple flower allele and a white flower allele. That's the F1 generation. And then if these organisms self-pollinate, because they're flowers, they can do that. They can have sexy times with themselves, but not asexual. Sexual, because they are producing, with meiosis, sperm and eggs, creating different options for the children. Then because each of these flowers could pass on a purple or a white flower gene, in this case, because of what we'll learn with the prediction, you actually end up with a ratio of 75% purple to 25% white, meaning the chances of their children being white are 25% chance versus the chances of their children being purple being 75% chance for at least the phenotype. And then the genotype's a little bit more complicated than that, but still predictable. All right, Punnett squares simplify this prediction process because it's letting us create those percentages and those ratios that will give us the information. So we know if we self-fertilize the plant, the sperm can be big P or little p, and the egg can be big P or little p. So for each parent, we put them on opposite sides of the Punnett square. So in this case, we have the sperm could be either big P or little p because it's heterozygote, and the egg could be big P or little p because it's heterozygote. And then if we do the cross between the ways they meet up, we can see big P and big P in this child, big P and little p in this child. And just a heads up, again, you always want to put the allele that's going to show up in the child first, because dominant always shows up. We always want the capital letter to go first if it's a mixed allele child. All right, this one is big P, and this is little p, and we do the analysis. All these guys are purple, and this guy is white, okay? And if we analyze the ratio, then we go, because it's out of four different ways these options can mix up, then we realize it's 75%, 3 out of 4 being purple, 1 out of 4 being white. If we were to analyze their genotypes, then I would say it's 2 out of 4 or 50% chance of being heterozygous, if it's big P, little p I'm looking for. All right, and just a reminder, genotypes and phenotypes will vary in terms of ratio. If I were to analyze the phenotype, I could look for homozygous dominant or heterozygote for chances of being purple, which is a 75% chance. And on the other hand, if I'm looking at the genotype versus homozygous dominant or heterozygote, which is showing the dominant trait, but they're not only dominant alleles, then I have a 25% chance of being homozygous dominant and a 50% chance of being heterozygote and a 25% chance of being white. Sometimes people do ratios. Ratios is the the comparison of the numbers. So it would be 1 purple or 1 big P to 2 big P little p's to 1 little p for the ratio. And for phenotypes, it is 3 purple to 1 white. I actually don't like the number system. I would usually, if you're doing a ratio, write down the number and then what that number is representing. So 1 big P, 2 big P, 1 little p when it comes to ratios. Now, the reason why this is allowed is because Mendel's first law of heredity, which is the law of segregation, which is the idea that each allele for a trait is packaged into a separate gamete. So if this organism is big P, there's a 50% shot of this of the allele that passes on being big P or big P, which in this case is 100% chance because they only have big P genes. If there are 2 alleles are little p, there's 50% chance of passing on this little p, 50% chance of passing on this little p, but because they're both little p's, it's always going to be 100% chance of this organism passing on little p genes. Well, in this organism that is big P, little p, there is a 50% chance of them passing on big P and a 50% chance of passing on little p, and that's being represented by that Punnett square. So if we look at the Punnett square here, the Punnett square justifies the probability because you're showing, hey, there's a 1 out of 2 chance of big P being passed on, a 1 out of 2 chance of little p being passed on, okay? And if you even run the numbers here, because there's a 50% chance of this big P and this big P getting together, that's 50% times 50% is 25% or 1/4. Oh yeah, it makes sense. 1/4 of my squares is big P, okay? So the law of segregation basically justifies, it's the idea that each allele is individually separated and randomly separated, and you're going to get a 50-50 shot of that allele being passed on. It's actually due to metaphase 1, because when you're deciding which of the 2 homologous chromosomes end up in a given sperm or egg, you really don't know which one it's going to be. Maybe it's the father on this side, maybe it's the maternal inherited chromosome on this side. You don't know. It's a 50-50 chance on whichever homologous chromosome winds up in a sperm or egg based on how they end up at metaphase 1 in meiosis. But either way, it's a 50-50 shot. That's being represented by the Punnett square. Now, this is helpful. These Punnett squares are helpful with things like test cross. So why would we want to use a test cross? Sometimes you'll know the phenotype of an organism, but you won't be 100% sure of the genotype. And this only really occurs with what we know as a dominant phenotype, because if we see the recessive phenotype, we know 100% because it only could have recessive alleles to show the recessive trait. It has to be little p, little a, whatever the little allele or weak allele is. But for the dominant phenotype, you only need 1 copy of the dominant allele. So I don't know if it's heterozygous for this dominant allele or homozygous for this dominant allele. And that's where a test cross would come in. because we know there's a 50% chance of either allele that this organism has being passed on to the child. So in this case, if I breed it with a homozygous recessive organism, I know, with the cross at least, that because we always know that one parent is providing the known parent, the homozygous recessive parent is providing a homozygous recessive allele, then we know that for the unknown dominant phenotype parent, that if they are homozygous dominant, it'd be 100% chance, 50% chance of passing on purple, 50% chance of passing on purple. It's 100% chance that that parent will pass on purple, and through the test cross, we now know that the outcome of this test cross, if the parent, the unknown parent, was homozygous dominant, all the children have to be homozygous dominant. But if the unknown parent was heterozygous for this dominant trait, then we know that if a white homozygous recessive kid shows up, that must have meant that this parent had to have at least one recessive allele, which means that this test cross lets us determine whether this unknown parent is heterozygous or homozygous. So that's a lot. So we're going to kind of like go through a step-by-step process to make sure we understand how to apply Punnett squares in the future problem sets. So in this case, remember, homozygous dominant means you have two copies of a dominant trait, and homozygous recessive means you have two copies of a recessive trait, and heterozygous means you have one different version for your two alleles, or one different version, two different versions of your alleles for that given trait, but we represent it using letters. So in this case, if I wanted to use any letter, I'll pick H in this case because the big H and little h look different. Then I can show it using big H to demonstrate it, big H little h to demonstrate heterozygous, or little h to demonstrate homozygous recessive. And this is just a showing we have a 50-50 chance of any allele from that parent being passed on to the child, right? So after meiosis is done, for this parent who's heterozygous with the big H and little h, there's 2 out of 4 or 50% chance of big H being passed on and 50% chance of little h being passed on to the child. So let's say we cross a parent who is, the dad is big H, little h, and the mom is 100% big H, meaning big H, and that's going to be passed on to the child. So in this cross, I am using the Punnett square. Ignore that. I'm going to use the Punnett square to represent the chances of each parent passing on their genes. So I'm going to pick this side for the mom and this side for the dad, but it doesn't matter. You could have put the dad's chances on this side and the mom's chances on this side. Now, because I know the mom has 100% chance, because both her genotype is big H, I know she has a 100% chance of passing on only the big H genes. I'm going to put big H on both of her two options. While the dad has a big H and little h genes, so he has 1 out of 2 chance of passing on big H, 1 out of 2 chance passing on little h. Now, I'm going to predict the kids using the Punnett square. In this case, big H from the dad, big H from the mom, it goes in like this. Now, just a reminder, dominant, the phenotype that's going to show up always goes first, okay? So in this case, it doesn't matter because it's only big H, so we don't have to worry about that. But over here, you might write it big H, little h. You might write it little h, big H. But that's a problem because the allele that's going to be the one showing up in the kid is the big H trait, the dominant trait. So this is not the correct way to write it because you might end up interpreting big H, little h and little h, big H as different, but they're still heterozygous. They have one copy of each. So we don't do that. We always write the big allele, the dominant allele first, for these two heterozygotes of big H, little h, and big H, little h here for this child. So that's the Punnett square on how we analyze genotypes and phenotypes. But this isn't actually the problem. Your problem is not likely going to be just make a Punnett square. It's probably asking you to do something else with it. Now, occasionally your problem will say, give me a Punnett square, but most of the times it's going to ask you something and you just use the Punnett square to do your prediction to do your work for showing what's happening. But the actual question might be something like analyze the offspring or analyze a cross. Or it might be something like provide the ratio of phenotypes and genotypes. So in this case, if you look at the predicted offspring here, you would see that 2 out of 4 are big H, and 2 out of 4 are big H, little h. So in terms of ratio, that would be 2 heterozygotes, or big H, little h, heterozygotes here, are there. And then there are 2 homozygous dominant, or big H. But like math, you want to reduce the number as much as possible. So the actual answer is 1 heterozygote to 1 homozygous dominant, or you can use the letters instead. So 1 big H, little h, the letter genotype, to 1 big H. Or if you want for your ratio, you can just use percentages or fractions. So 50% are big H, 50% are big H, little h for your genotype ratio. That's fine too. For your phenotype ratio, on the other hand, in this case, you might be tempted to write, oh, because all of them have the big H allele, they have curly hair, so 4 are curly haired. That's not a good ratio because a ratio always has to have a comparison. So in this case, we actually, because all of them have curly hair, we just say all are curly haired. Or if you want to use percentages, 100% are curly haired. Maybe your question is something else. Maybe it's like, what's the probability of the offspring being homozygous dominant? So in this case, when it's probability, you give a fraction or you give a percentage. So in this case, 2 out of 4 are homozygous dominant. So 2 out of 4 isn't a fraction. You want to reduce it. So 1 out of 2 or 1/2 are homozygous dominant, or you just give a percentage, 50%, because 1 out of 2 is 50% are homozygous dominant. So that's always what you want to do when you're solving these problems. Set up your Punnett square. That's your work. You always want to show your work and prove your point, and then answer the question. Now, do we have to use a square, 4 boxes for a Punnett square? Well, actually, not always, but this may be confusing for some of you. So I'm going to give you a tip that's going to be helpful if you understand it. But if you don't worry about it. You can just use the 4-box method. But in this case, you actually should realize that each side of the Punnett square is just representing the chances that the parent will pass on a given allele. Because the dad was big H, little h, he's going to have a chance of passing on a big H or a little h allele, 1 out of 2 of each chance. But because the mom is big H, homozygous dominant, in this case, she has a 100% chance of passing on the big H gene. So instead of needing to provide 2 options for the mother, because she only has the 1 option of passing on the big H allele to her child, you could just write big H once and you're done for the mom side of the square. And when you do the analysis here, with the 1 out of 2 chance of being big H and 1 out of 2 chance of being big H, little h for the outcome of the kid, it actually matches the Punnett square probability you got when you wrote all 4 boxes down and said, oh, because the mom's alleles are big H, this is the outcome. The boxes are just the sides of the Punnett square just represent the chances of the parent passing on the allele. And if the parent is only going to pass on 1 allele because they're homozygous for something, you can get away with just writing that letter once on one side of the Punnett square and all that. Now, like I said, you don't have to do this. You can just stick with the 4-box method. For most of you, that's going to be a lot easier. But this is a little trick for some of you who want to really simplify writing out Punnett squares as much as possible. All right, on that note, just remember, for solving our problem, we want to take the vocabulary we learned, translate our problem, make sure we understand what the genotype or phenotype of the parents are so that we can make our crosses and answer our question. And make sure when you write out your answers that you provide a Punnett square to show your work. That's your proof, that's your evidence. Even if you know how to get to the answer without a Punnett square, you're providing the Punnett square as evidence for your thesis. And in this case, your thesis or hypothesis is your answer for the problem, the percentage or whatever ratio you provide. All right, that concludes the lesson. We just basically went over how to apply all the vocabulary we learned to predicting monohybrid or simple crosses, and we understood why the law of segregation by Mendel allows us to use Punnett squares for prediction. And we didn't learn a whole lot of new vocabulary, but we learned how to use the vocabulary we learned in 13.1 to apply it to problem solving. Bye.